I'm a physicist by training, kind of an engineer by default, but a physicist by training. But I'm very, very interested in biology and how life works. And so my career has been an interesting story. I'm not going to get into that right now. But since I'm a physicist, I've taken everything I learned in my undergraduate and graduate and postdoc in physics and then translated it over to study biological molecules because I think they're fascinating. And I hope I can convince you of that today too. All right, so using the tools of physics to watch biomolecules work. Okay, so because many of you may not have taken biology since 9th grade, which was the last time that I took biology, I'm going to start at a pretty beginning phase, which is introducing the 3 main biological macromolecules, and those would be DNA, RNA, and proteins. And here I've selected different ways of representing the molecules. This is what they look like. They're huge classes of molecules, but these are representative pictures. And I hope you agree that they all look really different. And so I've picked these different representations to highlight that structurally, even though we think of them as the molecules of life, they all look different, they're made of different atoms, everything about them is different, and they do really, really, really different things. And that's what I'm going to try to hint at here, which is that DNA, again, was the one shown over here, it's like the reference volume. Okay, so I have been in Clark Hall since 1989, and at the time that I got here, we still had a library, and there was a reference section in that library, right? I see some of you nodding. You remember that? You would go into the library, and the books were too precious to come out of the library, so you had to look it up, copy what you wanted, and then take that out, to do whatever you wanted to do with it. DNA is like that reference volume, right? It contains all of the information for making the molecules that actually do things inside of your body, at least the ones that are manufactured inside of your body. And it's very, very precious. So it has to be kept, in this reference library where it can't be kind of tinkered with or changed, although sometimes that happens, right? And that can have deleterious effects. So DNA is all about information storage, and it was believed that all of the information in the DNA was used to make these molecules called proteins, which are the ones that it was believed that they carried out all of the different jobs that kept us alive. So if you're drinking coffee or you just had a donut, as I just did, there's proteins that are made to help you either digest it or just whatever it is. Proteins were thought to be the molecules that did everything. And RNA here in the middle was originally thought to be the messenger. So that was the entity that went into the reference library, figured out what proteins needed to be manufactured, copied the information out of the encyclopedia, and then took it to a place outside of the reference library where it could be acted on and then made into what was believed to be these functional molecules over here. So that was the view of how these molecules worked and interacted. We now know that that's not quite correct. I'll get more to that later, but for now, let's just keep it simple. So we have DNA, we have RNA, and we have proteins. And this is called the central dogma of molecular biology. DNA gets transcribed into RNA. It gets written or copied, and then the information gets translated or turned into proteins. Central dogma of biology. All right. So now, how do these molecules work? So I've told you a little bit, maybe in very loose terms, about how they're created. But, how does biology work? How do these molecules actually interact with each other, and do all the things that allow us to live? So it's actually quite interesting. There's a couple of factors, and this is just a nice illustration from a book that I use when I teach my biophysics class. So it turns out that these molecules all kind of fold up and have different shapes. So that was the point that I made in my earlier slide where I just showed you these different representations, and you're looking at it going, well, there's a protein that looks like a donut, that one looks like a stick, that one looks like a ball. What's the deal? Well, these molecules fold into different shapes. So here's one possible molecule with a shape that it's folded into. And then the cell is a really crowded place. There's a lot of molecules in there. It's actually kind of astonishing when you look at a representation of what a cell looks like at the molecular level. It's like a traffic jam. There's things in there, and they're all moving around and jiggling. And the way biology works, and this was a little hard for me to deal with when I first started learning about it, is that there's a lot of random collisions because everything is just moving around. And so this molecule A kind of moves around and has all these collisions and makes bonds with all of these other molecules that are there. And because things are moving around so much, if the bonds aren't tight, they come apart. But the one that it's supposed to bond to, right, the one where you have these molecules come together and then they actually have a function and more molecules come in, that happens because these two have exactly complementary shapes, like puzzle pieces. And so when they fit together, the fit is perfect. And so they're kind of immune to the kind of wiggling and falling apart that would happen if you had mismatched or loosely matched puzzle pieces. And in fact, that's a lot of how biology works. So first of all, and this is what got me interested in molecular biology, how do they know how to get to their shape, right? These molecules are all manufactured as long chains, right? That's called a polymer, a molecule that has a specific sequence of building blocks on it. And somehow that linear molecule knows how, right? I'm anthropomorphizing a little bit, but it folds up into the shape of a puzzle piece. All these other molecules fold into puzzle pieces, and then they can start the whole game of which ones fit together and work and do things like that. So they fold to get this particular shape that allows them to do their jobs. But what many people have been wondering about is, how do they know how to fold, right? I mean, actually, it's very interesting that you can take a beaker on a counter and put some of these the molecules in their linear forms, and they will actually fold into the right shape without the cell, right? They can do that right on the counter in your lab if you put them under the right conditions. So somehow, the information for what their structure is encoded in the sequence of building blocks that they're made from. All right, I'm not going to go into too much. You guys can take my class if you want to learn about the building blocks. That's about the first couple weeks of that class, and they're super interesting. But anyway, the molecules kind of know how to fold, and the information is somehow encoded in the molecule itself. But once it's folded, we're really interested in what does it interact with, right? So what are the partners that come together? Because a lot of biological function involves, tens, maybe sometimes hundreds of different partners coming together to do a particular function, right? It's very, very complicated. But how are the partners selected? What is the goal of the interaction? Why, when you have these two things come together, do they do something different than either one alone? So these are the big questions that will allow us to understand how life works at the molecular level, which I think we really, we don't know that much about it. We know something, but we're a long way from seeing the whole picture. Okay. So now, but wait a minute, I'm a physicist, right? And I know I've been talking about biology for the last 7 minutes. So how can I contribute? So physics or physicists like to observe. We like to make measurements. And then we develop and test models. And we say, these things come together. Gee, it looks like this and this are interacting. What if I change this? So that's the whole thing about being a physicist, right? You want to develop things, you want to build instruments for experimentalists that let you test things, and so that's what I do. And so I decided to try to build instruments that would let me understand the fundamental interactions that drive biology. So what I mean here is, what is it that allows a molecule to fold? How is this information encoded in this linear kind of worm-like molecule that eventually allows it to get this very particular shape? And then how do they act together, right? So what's going on the molecular level? There's, things happening. So what I like to do is to develop new tools and then use them to allow us to understand the physics that allows biology to work, right? I want to understand when this molecule folds up, I want to write down an equation that describes how this happens, and then I want to be able to test it and see if what I've learned in one molecule can be applied to another molecule, right? So that's kind of the goal of doing this. And also, I'm also very interested in computation and computational tools, but that's way outside. I mean, this could be a class. This is basically the outline of my class. But what I'm going to do is I'm going to focus on the tools that we've built that help us try to understand how these puzzle pieces form and how they interact with each other. So that's the goal of all this. All right. Moving along. Oh, yeah. So the first thing you have to ask, right, any self-respecting physicist would say, okay, what do I have to measure? I mean, would a meter stick do it, right? And, the answer is no, because these molecules are small. So you have to set the spatial, the length, and the time scales that you're interested in, right? So if I'm interested in, a car moving, maybe a meter stick and a stopwatch would be good. But what are the relevant length and time scales for biological reactions? And so these structures that I showed you, the protein shapes, the RNA shapes, the DNA shapes, they have interesting features on lengths from the atomic, right? Remember, hydrogen atoms, the smallest atom. Okay, the size of a hydrogen atom is about 1 angstrom. 1 angstrom is 10 to the minus 10th of a meter. So that means that you could have 10 to the 10 atoms, along my meter stick. 10 to the 10, that's, 1,000,000, you get the idea. So that gives you an idea of how small a single atom is. So because a lot of these processes, a lot of the reactions involve single atoms, even single parts of atoms moving around, we need to be able to measure on length scales from the atomic all the way through the overall size of the molecule. If I'm interested in how this kind of unstructured linear polymer folds up, I have to be able to measure its size when it's unfolded or when it's folded. So we need tools that can measure, so it's about angstroms through hundreds of angstroms, right? Again, this might be a new kind of realm for you, but think about 100 atoms back to back, kind of that size. Still, much, 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 much less than anything we can see with our eyes. Okay. And for time scales, well, it depends on what you're interested in. So here's a figure that I really like that just shows that the time scales for motions, can range from the second, right? So we all know 1 second here. A millisecond is 1,000th of a second. A microsecond is 10 to the minus 6th of a second. So in the time that I do that, if you can count to 1,000,000, right, that's a microsecond, right, 10 to the 6th. So pretty fast. And then these are even faster and faster and faster times. But fortunately for me, the things that I'm interested in are all relatively slow on the scale of the time scale that chemistry works on. This is all, chemistry is very, very fast, having electrons and parts of atoms move around. A lot of people work on that. My interest is up here in the second, millisecond, microsecond range. And just to give you a sense for what that means, everybody's blinking their eyes. That's about 100 milliseconds, 0.1 second. That's how long it takes you to blink your eyes. All right, so that's still up here, so we have to go maybe 1,000 times faster than that, maybe 10,000 times faster than that. So these are really short time scales. Okay, So many problems are on the millisecond time scale. So what I'm going to do is tell you two stories today about tools that we've built that allow us to measure, and I'm going to talk about two different biomolecules. Again, we're not going to go into the details, although I'd be happy to do that if you have questions. These are general stories. So a protein, right, that's already folded, so it's the puzzle piece is already formed, but we want to watch it interact with its partner. And the story I'm going to tell you, it's kind of interesting. It's about antibiotic resistance. So I'm sure you guys have heard about that some of these bacteria are becoming resistant to, you know, penicillins and things like that. We want to understand why that is. And so it turns out there's certain proteins that are designed to, you know, identify penicillin and just attack it and chew it up and inactivate it before the penicillin can kill the bacterium. Okay, so that's an example of, you know, I think that's a pretty relevant problem, you know, and then it can maybe help us if we understand why that protein chews up the antibiotic. Maybe we can design, chemically synthesize antibiotics that don't get chewed up and engineer new antibiotics that are effective at, you know, killing disease-laden bacteria. So that's one story I'm going to tell you. And the other one is I'm going to tell you about an RNA, right? So I love RNA. This is where my, you know, most of my research is in the nucleic acids. And people know very little about RNA because a long time ago when that central dogma was made, they thought it was just a boring intermediate and it just went into the library, did the copying, came out, and then self-destructed. But now, I mean, look at the Nobel Prizes. Nobel Prize was just awarded for, you know, another discovery about RNA last month. So with time, people have started to realize, ooh, this is an interesting molecule, and very little is known about it because most people have worked on, you know, proteins which are maybe more glamorous, elegant, you know, whatever you want to call them. So we are really starting our studies of RNA structure and how RNAs fold up and actually do their jobs. So anyway, so I'll tell you two stories that span these. They're on very different length and time scales. All right, so we build tools to measure these motions. So I want to watch these molecules work. Okay, I want to see this protein sit there. I want to watch the penicillin go into the binding site, and I want to watch the protein chew it up and understand how that happens. So we build tools that let us watch things happen in real time, and we use microfluidics. So because the NanoFab facility is here, CNF, right, natural to use to fabricate, you know, make devices. And so we have all kinds of little devices that allow us to mix things together quickly and then, you know, and then watch them work. And then we use X-rays to let us see, right? So light has wavelengths that are, you know, about a micron, okay, 10 to the minus 6 of a meter. So using light, we can see things that are about the size of the wavelength. So since we're down at 10 to the minus 10th of a meter, we need light with a much shorter wavelength, and that would be X-rays. X-rays have angstrom wavelength. You can see things with angstrom size using X-rays, okay? You know, not exactly in the same way as we can see things with micron size using light. But just, you know, for that, to give you a sense of why we can't use light, just that it's not the right match to the size of the system. And again, we're really fortunate to have X-ray sources even here on campus. So we use CHESS a lot, our local synchrotron. Okay, so we use microfluidics that, you know, we make a lot of them at CNF, and we've started our X-ray program at CHESS, but we've expanded way beyond that. So very local. This was a very homebrew kind of project. All right, so now I'm going to start telling you about some of the details. So everybody good with the, you know, kind of understanding what we're after? We want to watch these things react, all right? And then we want to see how they move so that we understand at the atomic scale how, you know, antibiotics get chewed up, how RNAs, get to assume the pieces, the shapes of these puzzle pieces. So the trick here is a device, a microfluidic device that I made the first ones myself when I was a postdoc in Saul Gruner's group here at Cornell in collaboration with Bob Austin, who was at Princeton. And they're actually incredibly simple devices, but you have to know a little bit about physics to understand how they work, and I'll tell you, it's not hard. So these devices are 2 cross channels, and we go over to CNF, and we just etch this plus sign all the way through a silicon wafer. So when you hold it up to the light, you see a plus sign in it, right? That's what we're doing, although it's a lot smaller. So the width of the channels is about 50 microns over here, and depending on the weather, that's about the width of one of my hairs. Okay, so think about it. This is about the width, like 2 pieces of hair crossed together. That's the scale of this device, all right? And then we seal it up, we put windows on the front and back, and we drill holes. And then we can flow fluids inside these channels that are the width of one of my hairs, right? It's called microfluidics. People do this all the time. And what happens is we put a big molecule in one kind of solution surrounded. These are all molecules that float in a buffer. We could call it water. It's not really water, but we could call it water to keep it from being confusing. So we put a large molecule in water in one solution, and we flow it in through this port over here. So the liquid's always flowing left to right. And then we put in another liquid from the top, the north and south channels here. And this solution could be water or buffer, but it contains one component that's a little bit different than this solution A. There's one thing that's different between solution A and solution B. This is where the physics comes in. There's something called diffusion. And so diffusion is just the way that things move around because they have some energy. It turns out that if you have a big object like a protein, it doesn't diffuse very far because it's big. It has to push a lot of stuff out of the way. But if you have a tiny object like a salt ion or maybe an antibiotic, right, which is penicillin, which is a lot smaller than a protein, that zips around a lot faster, okay? So this other solution hydrodynamically focuses. It just means that we push harder from above and below, and we squeeze this solution into a really thin jet, and it's all flowing out to the right here, okay? And because these little small things are present in B but absent from A, they will diffuse very quickly across this green channel, and they'll diffuse into this. But the big molecules, because they don't diffuse, they will just follow the flow lines here. They won't move up or down. They'll just go straight. So this is how we mix things together. We mix a small thing with a big thing. And then what happens is we have our big thing coming in over here, and the small thing diffuses in here. And then if the big thing wants to act on the small thing, right, think about a protein and an antibiotic that it's going to chew up. I'm really giving away the story here, but it's okay. Then we can actually say that we've turned space into time, right? This is the big thing. And this point in space is a fixed time after the 2 molecules met. And so this is one time point at this location in space, and things flow down, and here's another time point at this location in space. And so I'll tell you this, when my girls were little, they took gymnastics every Saturday morning, and I would go down to the gym with them. And there would be a big mat, right? And all the kids would run down the mat. And there were points where they would have to do a cartwheel and all this. So think about this. These molecules are running down the mat, and we are just sitting. We've pulled up a chair at one position. Maybe all the kids are doing cartwheels, whatever it is. And we're just watching all of the different cartwheels right here for however long it takes until we understand. So if this is happening, even if it takes a millisecond for the fluid to flow from here to here, we can sit here as long as we want and just always measure what's happening at that particular time in the reaction. And then we pick up our chair and we move to another location, and that's how we map out the entire time course of a reaction. We put some kind of an external probe right here and we watch what happens, okay? Yeah, okay. So now we're going to talk about the probe that we use, and that's X-rays. So we basically put X-ray beams at different locations along that outlet channel, and we measure, because X-rays can see angstroms, we can measure structural changes on the angstrom scale. And so there's really two standard ways that X-rays are used to measure biomolecules. Unfortunately, it's not as easy as, just light and looking at something and saying that's where it is. It's a little bit more complicated. So we use either crystallography or something called solution scattering. And I'll tell you what both of those means. So we are going to start with crystallography. All right. So surprisingly, and this was a surprise to me, you can actually, let me just advance here. So I'm going to tell you more about X-ray sources in the next couple of slides. But you can make a crystal out of biological molecules. This really surprised me. So, I have a little tiny diamond on my ring over here. And a diamond is a crystal that's made out of carbon atoms. And that just means that the atoms are arranged in a regular way in space, right? And you get a crystal. You can get salt crystals, sugar crystals, things like that. Well, it turns out you can actually make a crystal out of proteins. People have been doing this for a long time. And here's some proteins, and they're arranged, like eggs in an egg carton, right? Three-dimensional egg carton, and you get a protein crystal. And when you shine X-rays onto your protein crystal, because everything's so ordered and organized, you get reflections, right? You just get, there's interference between atoms that are at the same place in the different locations, right? Just take my word for it. You get these spots. I mean, it's probably a much longer explanation. That's a full lecture in my class. I'm not going to do that in 5 minutes. But you get these spots over here, and they're really well organized, and they're really beautiful. And by analyzing those spots, you can actually figure out what it was that's inside of this protein crystal. And we draw proteins like this because they have the beautiful structures and shapes. But I want to remind you that they're actually, this is like looking at the strand of a necklace. There's actually beads, right? So if I just look at the thread that runs through the beads on a necklace, it could trace something like that. But this adds all of the atoms. They're really very, very beautiful and tightly packed structures, and this is about an angstrom. These are individual atoms. So from these X-ray crystallography, you can actually figure out what the structure of the protein is to angstrom resolution. Here's a hydrogen atom. Here's a carbon atom. Here's an oxygen atom. And it's really remarkable, and we've learned a lot. The problem with crystallography is that you have to keep everything frozen in this crystal. And so you can't measure big motions because it's all frozen. If everybody's sitting in seats in a room and somebody starts moving around, everybody's going to get up and leave. It's going to disrupt it, okay? So that's the downside. You get very high resolution, but you don't have a lot of motion. All right. On the other hand, what I have really spent most of my career doing is something called solution scattering, where instead of having the atoms in a crystal, we have them kind of dissolved in liquid. So, if you have a sugar cube, and you put it into a container on the counter, it'll dissolve. So you can think of the crystal, kind of like being that cube form. This has everything dissolved, and the proteins are all floating around. And what I like about this is because they're floating around, they don't have this problem. They can take any shape they want. They're not going to melt, right? So they're allowed to sample all kinds of shapes, but you lose the sharpness with which you can detect the features. And so this is a protein we've worked on, and my crystallography colleagues would be able to tell you something. It looks beautiful. This is the backbone, kind of the thread that holds the beads together. And what I would measure is something that looks like that, right? So there's like a blob with something hanging off the side. So you trade the very high sharp resolution for the ability to measure large motions. And they're both valuable. So, actually I'm going slowly, so I'm just going to skip this last part. I think it's not important. So, okay, how do we do the measurements? So remember, I'm a physicist and I'm all about doing measurements. It's really simple. So we have an X-ray beam coming in just like we did for crystals. But now we have our liquid sample, our sugar that's dissolved. So the molecules are all kind of floating around in there. And then we have the straight beam comes in and it scatters, right? Because the molecules disrupt the beam. It's straight, it hits my molecules and it goes in all kinds of directions. We collect the scattering on a detector that's very much like the camera in all of our cell phones. So there was a time when my cell phone was probably more sophisticated than the camera that we were using, just because they have some other issues, but it's very funny. These same kind of camera where you can get a picture and you can actually see this is where, this just shows that's where the beam comes in, and this bright red is the high intensity, and it just kind of falls off with distance, and we just basically measure how the scattering changes across the detector here, okay? And the only thing, and I didn't want to get too technical here, but there is one thing that we have to know, which is that we usually measure in terms of the angle. So the straight beam comes in and we measure how many X-rays are scattered to this angle, which is a cone, and then how many to this angle, which is a cone, and to this angle. And that's really important because this is plotted. Then you put that intensity on a graph, and the x-axis is in inverse distance. And skipping a whole lot of stuff here, 2 lectures in my class, what it means is that the information out here at low Q tells us about big distances. This is small 1 over distance here. This is large 1 over distance here. So we get information about big distances at the small Q and information about small distances at large Q. And don't worry about understanding that, but that's going to be important because we're now pushing to measure out very far and get information about the shortest distances we can. Okay. All right, so where do our X-rays come from? Well, we have a lab source, okay, downstairs, a wonderful machine. There's about 5 X-ray machines in the physical sciences building complex, right? You can buy them. They're all controlled. The state regulates everything. The rooms are locked. We have, all the safeties in place. They're not hazardous at all. But there's 5 of them in this building, okay? Synchrotrons are national facilities, right? They are much bigger sources, and they provide really different kinds of X-rays. There's about 5 of them, 5 of them in the United States, including one right here, CHESS, our local synchrotron. So going from these home sources, and there's a bunch in this building, now there's about the same number in the United States. But what I've been really excited about are the latest generation of X-ray sources called X-ray free electron lasers. And there's 5 in the world. Okay, so and each one of them brings us different types of X-rays and allows us to do different types of work. And that's what, as a physicist, I can say, I can get this kind of X-ray from this machine. I know that X-rays can tell me about the atoms inside of proteins or RNAs. Which one do I want to pick? What am I interested in? Which source do I want? And so that's what I do. So right, the X-rays from each source have distinct properties. And just to try to give you some context, this is a comparison, not to size, right? Not to size. But my source downstairs creates an X-ray beam that's a couple millimeters in diameter, and we get 10 to the 8th X-rays per second. So if I was just with a catcher's mitt catching and counting the number of X-rays that my home source made every second, I would count up to 10 to the 8th, right? That's 1, 8 zeros after it. At chess, I get 10 to the 12th photons per second, 10,000 times more X-rays, and that's a smaller beam, right? You know, kind of the relative size is right. But at the XFEL, I get 1,000,000 times more X-rays per second than I get at my home source, and I get it in that teeny tiny dot, right? So you can see that you know, things really change a lot depending on what you're doing. So just to give you maybe a better feel for it, if you think of a lab source as having roughly the intensity of a flashlight, then a synchrotron would be like a laser, right? So it's more intense and it's more tightly focused. And then going from a synchrotron to an XFEL, these X-ray free electron lasers send pulses of X-rays. They're not a continuous beam, really. You know, I mean, synchrotrons aren't continuous either, but they come so quickly you think they're continuous. X-rays, it's like pitching. You know, so I used to play tennis, and there was a ball server that would just pitch a ball at me, and I would practice with it. That's what it's like. We get these pulses of X-rays that get shot at our sample, and they're super short. I mean, femtosecond, 10 to the minus 15th of a second, super short. They come, there's 120 per second, and each one of these pulses has as many X-rays in it as 1 second of beam at chess. So if I sat at chess with my catcher's mitt, I would catch in 1 second 10 to the 12th X-rays. Each one of these balls has that many X-rays. And so what we can measure in 1 second at chess, we can measure in 40 femtoseconds, like 10 to the 15th times faster at the X-ray free electron laser, just a whole lot faster. So, and interestingly, we can actually get enough X-rays in here to measure a diffraction pattern, get all the spots, right? So one pulse gives us enough information to see all the spots. All right, so this is what XFELs look like. We use the one at Stanford, right? So aerial view, it's I think a couple miles long over here, all the way down, you know, make all the X-rays, condition it, the whole thing. And we work in this little tiny hutch down here. And I will tell you that oftentimes we are the only users of this facility. And it's a little bit, you know, because everything is so conditioned. So they will make the beam and it comes and it is delivered just for us. And all these people are working and keeping things controlled so we can do our experiment. So it's really hard to get. You have to have a really good case because there's not a lot of beam time. They're making it so that more than one person can work at a time. But we take this really seriously. And sometimes if we don't like things down there, we'll call these guys and say, hey, could you move it to the left, you know, just a little bit? And they'll do it because, you know, they're making the X-rays for us. So this is a very serious responsibility, taxpayer funded. We take this very seriously, and when we go out, we are prepared, and we know what we need to do. We've got all our ducks in a row, and we do not waste a minute. Okay. So what do XFELs do for us? New opportunities afforded by XFELs. So because they're X-rays, we can do crystallography, and we can do solution scattering. That doesn't depend. You know, I could do both of them in my lab downstairs because we get the same opportunities for spatial resolution. But what this huge flux and the small size of the beam, it gives us new opportunities to do better time-resolved measurements, right? To watch things happen in time, to use our mixers in a different way so that we can actually look at these millisecond to microsecond scale events in these samples. All right, but there's challenges, right? We got to talk about this. And the first one is that I told you that each one of those balls coming down, like those tennis balls, they're really powerful. Yeah, they blow your sample up. So every time one of those powerful X-ray pulses come down, there's 120 per second. And they're so fast, but they hit the sample and they blow up everything in their path. Okay, so you would think that would be a showstopper, but it's not, because people said, hey, we'll work with it. And so they did, some people who are now collaborators of mine, but about more than a decade ago, they decided we could get around this because you inject your samples using something like a nanoscale garden hose. So you have a little hose that shoots your molecules, whatever sample you're looking at, it shoots it into the beam so that when it shoots it in, right, it comes out, it's a free jet. You get one little crystal or whatever it is here, the beam hits it, it diffracts, and then it blows up. So what? The pieces fly, and then another one comes in. So this is called diffract before destroy, and in fact it works. And about that time, a colleague who was visiting me at Cornell said, ooh, what if you put your flow cell here, and we've been building these things with these liquid jets, right, that spray into the beam. You have this microfluidic mixer that you've been using at chess and all these other sources. What if we put our heads together and find a way to put your mixer in there? So that's what we did. We got an NSF Science and Technology Center called BioXFEL that had a whole lot of people in it, and I was just one component of it. But it ran for 10 years, and it allowed us to actually build the technology to do this. Okay, so I have two applications, but I'm way over on time. I think I'll just talk about the first one, right? Because, I respect your time. I could go on, but, so we'll just talk about the first one, but knowing that there's a second one. But the first one, we contributed to just mainly in the technology. So we measured angstrom scale changes in proteins. So this gets back to the antibiotic resistance problem. So we now have our mixers working at the XFEL. We can do both crystallography, and we're learning how to do solution scattering. It's very hard for a lot of technical reasons, but we'll figure it out, right? We're getting there. But I'll tell you about the crystallography story, because I think that's interesting. So yeah, remember, we have lots and lots of X-rays in a teeny tiny beam. So in about 2012, when this machine first came online, a whole lot of people who were not colleagues of mine then, but are now colleagues of mine, used these nano garden hoses to shoot individual crystals through the beam where they were struck by a pulse and made a diffraction pattern. So one pulse, one crystal, one pattern, kaboom. That's the way it goes, right? And then you get another pulse, another crystal, another pattern, kaboom. And then you get all the patterns together. And it turns out that in standard crystallography, what I was telling you about before with that big protein crystal, you actually have to look at the full three-dimensional. You can't get the structure from one snapshot. It's a three-dimensional object. So you take that big crystal and rotate it, and get structures everywhere through it. Some very smart people figured out that if you have tens of thousands of tiny crystals in random orientations, you get the same information as you have from one big thing that you rotate. You just have to do some work to put it all together. But they've done it, and now we can get structures out of, a couple thousand crystals like this. Okay, so this actually is a new opportunity because, the one other thing is that these crystals are tiny because why would you build a huge crystal and then blow it up? You know what I mean? So you want to make something that's about the size of the beam so that you don't have to use infinite amounts of sample. It's going to blow up anyway. Make a small thing that's going to blow up, and whatever, make a lot of small things. But now that you have small things there, you remember what I told you before about diffusion. So if you have something with a small length scale, you can actually get very small atoms or molecules to diffuse through it. And so there was this idea that we could actually have a crystal of proteins, right? And then flow things in that would diffuse into the crystal, because the crystal is mostly water, interact with the proteins in the crystal, and then if the motions are small, we can watch the protein actually doing its thing and working on those molecules. So our goal was to measure protein dynamics, looking at the proteins locked in this crystal. Remember, they can't move very far, but a lot of these reactions, there's a lot of chemistry. They don't have to move for these. You can do things with small reactions. And so this is what we set out to do. So here's my mixer. Here's this nanoscale garden hose. And about 10 years ago, I had a very, very brave student. And I said to him, fit this into that, right? So this is your thesis project, right? Just make that work. And he actually did it, all right, with help with other people. And so we did that. We could now mix things together. And as part of this NSF Science and Technology Center, we had some people with interesting biological problems. I mean, you can't do them all. It's very hard to do all the technology and be breaking new ground with the technology and also be breaking new ground on the biology. You need collaboration. That's why people work in these big teams. But the problem that one of our collaborators was working on was antibiotic resistance, which I think I've mentioned a couple of times. And the idea is that there are these antibiotics like penicillin that are widely used to treat bacterial infections. Some bacteria have acquired a protein. So they got the gene, the DNA, the instructions for making this protein. And this protein actually kills the antibiotic before the antibiotic can kill the bacteria. It's a good strategy if you're a bacteria. And people kind of thought they knew how this worked, but we wanted to watch it, right? We wanted to see the protein going in and working. And so these are slides from my student Andrea Katz, who I just love the way she did this. Here's a bacterium. Here's the protein, this beta-lactamase, it's what it's called, inside of the bacterium. Here's the antibiotic, right? So this comes in, gets inside, and then it does destructive things and it blows the bacterium up, all right? And so the way this works is that the first thing is the protein actually grabs on to the antibiotic. This is the enzyme substrate complex. And then it was believed that it breaks this ring over here. It kind of grabs it and opens this up, and then it destroys it, right? So it uses some kind of chemical reaction to break that bond, and then it releases that one, and it lies in wait for the next antibiotic to come in. So this antibiotic loses its potency once that ring is opened up, right? It's called beta-lactam, whatever. That's another whole story. But the point is that this opens it up, it loses the potency, and then it releases it so that it can lie in wait for the next penicillin to come in. So one protein can just do this over and over and over again, and it's actually fairly effective for the bacterium. So this is actually a picture of the mixer that my students built to do the experiment. It's really, 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 it's just a beautiful, high-tech, cleverly engineered piece of equipment. We worked in collaboration with Chris Schaefer's group over in biomedical engineering. They helped us actually laser machine some of the critical components that we needed to do this. But here you can see there's our mixer inside. It's all sheathed in this larger glass capillary. It's concentric glass capillaries. But the point is, now we put a protein crystal inside here. So this is our large object, and it gets focused into a stream. And then the antibiotic goes in around here. That diffuses into the crystal, into the stream, into the crystal. And then down here, it gets spat out where we shoot it with the X-FEL beam. And we get some information about it. And so I really don't want to go too much into the data, right? But we do have data over here. And you can see the resolution. But I do want to point out just the highlights. Over here, we captured the time at which this antibiotic was captured by the protein. And you can see here, that's the antibiotic. And the distance between this point over here and this arm of the protein is 3 angstroms. And in the next time point, we can actually see that that distance has shrunk to 1.6 angstroms. So we watched a covalent bond form, right? So the resolution of this measurement, it's so good that we can tell when two atoms are close to each other and when the bond is formed. So it's really, and you need angstrom resolution. And the formation and destruction of bonds, that's how life works. It's all really chemistry. Chemistry, physics, biology, it's all the same thing. It doesn't matter. That's how it works. Okay, so I just went through about 4 papers worth of stuff here. There's a lot more, but just to give you a sense, and I'm not, unfortunately, going to get to the second story, but we demonstrated that we can actually watch proteins work with angstrom resolution in crystals. And actually, most recently, my most senior student right now has now found a way to allow us to do this at synchrotrons. Remember, there were only 5 XFELs in the world, and it's one at a time, right? Like one experiment at a time, getting better now, but the time is so hard to get. It is so competitive. There's a lot more synchrotrons, and they can have hundreds of users together. So we figured out how to take this technology and translate it to synchrotrons. And in her most recent paper, which is submitted, it's being reviewed right now, we actually resolved a controversy. There was a protein, and people argued, it does this, and the arguments go back and forth in the literature. And we said, we'll just look, right? Because everybody's making a secondary judgment. Oh, well, after the reaction, you see this. And we said, well, we'll just measure it. So we mix them together, and we can actually watch this molecule go in and bind in this certain way, and we can watch the reaction happen. So that's kind of pretty amazing, right? And that protein actually is implicated in human diseases like Parkinson's. So we are now moving towards, really trying to address problems of great biological interest, right? You have to develop the technology first and demonstrate it and test it out, but we want to do some significant biology. So now I'm going to skip through the whole second part, which breaks my heart a little bit because, this was the one, after we did the technology, I wanted to do my own science, which was with RNA, and we did it. But anyway, I think we're just going to skip through this whole part and all of this stuff, and I'm just going to go to my acknowledgments here. So X-rays plus microfluidics are really a unique tool for studying how biomolecules work. I told you the story about using crystallography with atomic resolution. We have this other story where we can watch large-scale changes, and I think that has potential for really opening up new ways of determining structure. And of course, I need to acknowledge everybody who did the work, right? So this focused more on the last part, but the point is that this is how many people it takes to do an experiment like that. And we're from all over the place. This is a pretty US-based collaboration. We do work in Europe sometimes in Japan, very international field. And there's my group from about a year ago, and the funding, of course, a lot of it, everything that I talked about came from the NSF Bio-XFEL Science and Technology Center. And the RNA part is funded by my NIH grant. Anyway, thank you, and I'm sorry I ran a little bit over, but I'll take questions.